inside the nucleus of all of our somatic cells. And when I say somatic cells, I mean all of the cells of our body that are not sex cells, not sperm or egg cells. But all the other cells in our body contain 23 pairs of chromosomes, which equals 46. Here is a chromosome, one of 46 in all of your cells. And a chromosome is a form of DNA when a cell is dividing. When a cell is not dividing, our DNA is in chromatin form, a long thread-like version of chromosomes. Just think of pulling a thread out of a sweater and how long it would be if you actually pulled the entire thread out of that sweater. If we zoom in even further, we see that our DNA is wrapped around proteins called histones. So one complex of DNA around a histone protein is called a nucleosome that we see here. And if we zoom in even further, we can see our pairing rules. A pairs with T's, C pairs with G's. Those are our nitrogen bases of DNA. You might recall from the previous chapter that DNA is nucleic acids, and the building blocks of nucleic acids are nucleotides. And a nucleotide consists of three parts: the sugar, the phosphate, and the nitrogen base, whether it's A, G, T, or C. So here we can see 1, 2, 3, and 4 nucleotides on this side of the DNA molecule. We can see that the nitrogen base is paired with its complement on the other side of the DNA molecule. Adenine always pairs with thymine, glycine always pairs with cytosine. The sugar used in DNA is deoxyribose. Just to reiterate the base pairing rules, adenine and thymine go together, cytosine and guanine go together. Here they are in molecular form. We can see the two sides of the molecules joined by hydrogen bonds. So if you can imagine, hydrogen bonds are the weakest type of bond, so it wouldn't be very hard to split this DNA molecule right down the center between those nitrogen bases. Before a somatic cell divides, it has to replicate. This slide is on DNA replication. That's the process of taking your 46 chromosomes and copying them. So when the cell divides into two cells, each new cell will have a complete set of 46 chromosomes. This way we conserve our genetic information as cells divide. Here's how DNA replication works. Well, our DNA molecule, which is a double helix, is unwound. This enzyme here, called helicase, unwinds the double helix. Do you see how it's taking the double helix and splitting it right between the nitrogen bases? So some nitrogen bases are on one side and the other ones are on the other side. Then nitrogen bases are added to each side by this enzyme that you see in yellow called DNA Polymerase. Poly means many. Ase is the ending for an enzyme, just like we see with helicase. So all of these nucleotides that are single-stranded now because helicase unwound it, well, DNA polymerase adds the nucleotides according to the pairing rules. So if it sees an A, it'll add a T. If it sees a C, it'll add a G. And then on this other strand, the same thing occurs with another DNA polymerase. This is called semi-conservative replication because each new DNA strand consists of one old strand and one new strand. One strand is called the leading strand, and one side is called the lagging strand because it can only replicate one way. The leading strand will follow helicase as it unwinds, so it never has to let go. The lagging strand is moving away from helicase, so by the time it gets to the end, helicase would have unwound further. Therefore, it has to detach and replicate over again in segments.